Hello students, welcome to EPG Pathsala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about a module on ATP generation under paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. We will try to understand the structure of ATP synthase. We will also try to understand the mechanism of synthesis of ATP and also understand the structure of component of electron transport chain. Adenosine triphosphate is a nucleoside triphosphate used in cell as a coenzyme, often called the molecular unit of currency of intracellular energy transfer. ATP was discovered in 1920 by Carl Lohmann and independently by Cyrus Fickes and Subbarao of Harvard Medical School. But its correct structure was not determined till some year later. It was proposed to be the intermediary molecule between energy yielding and energy requiring reaction in cell by Fritz Albert Lippmann in 1941. It was first artificially synthesized by Alexander Todd in 1948. ATP transports chemical energy within cell for metabolism. It is one of the end product of photophosphorylation, aerobic respiration and fermentation, and used by enzymes and structural protein in many cellular processes including biosynthetic reactions, motility and cell division. One molecule of adenosine triphosphate contains 3 phosphate groups and it is produced by a wide variety of enzymes including adenosine triphosphate synthase from adenosine diphosphate or adenosine monophosphate and various phosphate group donors, substrate level phosphorylation, oxidative phosphorylation in cellular respiration and photophosphorylation in photosynthesis are 3 main mechanisms of ATP biosynthesis. The structure of ATP molecule consists of a purine base, that is adenine, attached by 9th position on nitrogen base, and on 9th nitrogen atom to 1 carbon atom of a pentose sugar, that is ribose. 3 phosphate group are attached at 5th carbon atom of the pentose sugar. It is the addition and removal of this phosphate group that interconvert adenosine triphosphate, adenosine diphosphate and adenosine monophosphate. When adenosine triphosphate is used in DNA synthesis, the ribose sugar is first converted to deoxyribose by ribonucleotide reductase. Following figure shows the concept map of ATP generation. We know that ATP can be generated by 2 ways, by substrate level phosphorylation and ATP generation by electron transport chain. In metabolism, ATP is generated by 2 fundamental different biochemical mechanisms as mentioned: substrate level phosphorylation and ATP generation. In substrate level phosphorylation, ATP is formed from ADP by transfer of a high energy phosphate group from an intermediate of a fueling pathways. The reaction so as a, for example, as consequence of the removal of a molecule of water, the low energy ester linkage of phosphate in 2-phosphoglyceric acid is converted to high energy enol linkage in phosphoenol pyruvate as shown in figure. If you see in the figure, the high energy linked phosphate can then be transferred to ADP, the consequence of which is generation of a ATP molecule. Generation of ATP by electron transport chain. In a number of different modes of microbial metabolism, including respiration and photosynthesis, ATP is generated by transporting electron through the chain of carrier molecule with fixed orientation in a cell membrane. Although the complexity and component of electron transport chain vary, they have a certain common features. The component of chain are carrier molecule capable to undergoing reversible oxidation and reduction. Each member of chain is capable being reduced by reacting with the carrier molecule that precede it and oxidized by the carrier molecule that follow it. In any specific example of an electron transport chain, certain members transport hydrogen atoms while others transport only electron. The orientation of carrier in the cell membrane is such that hydrogen carrier transport in the direction towards the outside of the cell and electron carrier transport towards the inside. Thus, at each conjunction in the chain of a hydrogen carrier and an electron carrier, a proton is transported out of the cell. The membrane is otherwise impermeable to proton. As a consequence, electron transport trap a portion of chemical energy released by the net reaction on the chain in the form of gradient across the membrane of proton and electric charge. Such a gradient, termed as protomotive force, is form of potential energy capable of doing work. It drives certain permease system that concentrate externally supplied substrate within the cell. It provides the energy for flagellar mediated cell motility, and it drives the energy requiring synthesis of ATP from ADP. Figure shows the structure of ATP synthase. The ATP synthase, an enzyme that synthesizes ATP at the expense of protomotive force, is catalyzed by a complex membrane-bound enzyme, ATP phosphohydrolase, sometimes termed as ATPase, composed in all bacterial of two multi-component protein called BF0 and BF1. The subunit composition and membrane insertion of BF0 and BF1 are shown in figure. The alpha and beta subunit of BF1 are arranged alternatively to form a hollow hexagon, the central hole of which contains the gamma subunit associated with other subunit of sigma and epsilon. The BF1 probably has the subunit structures as shown in figure. The alpha and beta subunit from the catalytically active portion of structure, the site where adenosine triphosphate is synthesized from adenosine diphosphate, an inorganic phosphate. The gamma and sigma and epsilon subunit form a proton translocating stock and get that bring to the active site at the proper rate and proton that drive the reaction. The peptide form a proton channel through the membrane. They are highly hydrophobic, accounting for their intramembrane location. ATP phosphohydrolase catalyze a reversible reaction. ATP can be synthesized at an expense of proton motive force, or in certain cases, a proton motive force can be established at the expense of intracellular ATP. Now, if you see the role of ATP, ATP can be used in many ways. It is used in metabolism, it is used in synthesis and active transport process. It is also used and it has role in cell structure and locomotion. Extracellular signaling and intracellular cell signaling also require ATP. ATP is also required in DNA and RNA synthesis. Following figure shows the value for the components in electron transport chain. In order for an electron transport chain to function, there must be a gradient of susceptibility to oxidation. That is, each component must be capable of being reduced by reduced form of previous component and oxidized by the oxidized form of subsequent component in the chain. The relative susceptibility of a substance to oxidation or reduction can be described quantitatively in terms of its electrode potential or reduction potential. This is the relative voltage required to remove an electron from hydrogen. Thus, standard reduction potential is that the hydrogen electrode, which is assigned as an arbitrary value of 0 volt, and under standard condition at the pH 7 near which most biological reactions occur, the potential of hydrogen electrode is 0.42 volt. The symbol of E0 designate electrode potential measured under these conditions. Knowing the electrode potential value of two-half reaction, the free energy change of a coupled reaction can be calculated from the relationship. The free energy change at pH 7 is designated as G0 N is the number of electron transfer, F is the Faraday, and standard E0 is the Algren difference between the potential of two-half reactions. For a typical biological oxidation, for example, the oxygen-linked oxidation of NADH, the analogous calculations show a free energy change of 42,400 calories, not significantly different from the peroxidation of hydrogen. The carrier in an electron transport chain participate in a series of reactions of increasing values of electrode potential between that of the primary electron donor and terminal electron acceptor. The electrode potential position scale of several typical electron carrier, primarily electron donor and terminal electron acceptors, are shown in figure. Figure shows the component of an electron transport chain. The electron transport chain of aerobic chemoheterotrophs are the most thoroughly studied. Those involved in oxidation of organic compound always contain four different class of molecule. Two class, the flavoprotein and quinones, are hydrogen carrier. The other, the iron, sulfur, protein and cytochrome are electron carrier. Flavoprotein have a yellow colored prosthetic group derived from biosynthetically from flavin, that is vitamin B12. The prosthetic group may be either flavin mononucleotide, that is FAD. Both possess the same active site capable of undergoing reversible oxidation and reduction by donating or accepting two hydrogen atoms respectively. The flavoprotein are members of a large class and differ widely with respect to electrode potential. Some are active in the primary dehydrogenase of organic substrate, that is succinate. Others participate as hydrogen carrier within an electron transport chain, and still other electron transport chain contains either ubiquinone, substituted benzoquinone, or menaquinone, a substituted naphthaquinone. The former occurs mostly frequently in gram-negative bacteria and in the mitochondria of eukaryotes, and the latter most frequently in gram-positive bacteria. A few facultative anaerobes, including E. coli, contains both quinones but tend to use ubiquinone for aerobic respiration. On reduction, they accept two hydrogen atoms to form the corresponding quinol. Treat directly with molecular oxygen with formation of hydrogen peroxide. Following slide shows the various component of electron transport chain. Again, you can see the component of electron transport chain. The iron sulfur protein that contains 2, 4 or 8 atoms of labile sulfur, so called because they are released as H2S by strong acid. The iron atoms in 2-FeS protein are held in a lattice composed of 4 atoms of cysteine, sulfur, and the 2 labile sulfur atom. The iron atoms in the 4-Fe4S protein interact with 4 labile sulfur atoms to form a cube that is held in place with the protein by 4 cysteine residue. The 8-Fe8S protein contains 2 active centers identical to those found in 4-Fe4S protein, thus enabling them to accept on reduction 2 electrons, while 2-Fe2S and 4-Fe4S protein can accept only 1. The cytochrome belong to the class heme protein, a class that also include hemoglobin and catalase. All have one or more prosthetic groups derived from heme, a cyclic tetrapyrrole with an atoms of iron chelated within the ring system. There are 4 types of heme prosthetic groups known as A, B, C, and D that differ particularly in the nature of substituent group at the periphery of the heme. Individual cytochromes contains one or more heme groups in various combinations. The molecular weight and reduction potential of cytochrome vary widely. Cytochrome content of bacteria is quite variable among species and environments. For example, an aerobically grown bacteria will have a different cytochrome composition from that of the same strain grown anaerobically or even grown with a restricted supply of oxygen. The presence of A and C type cytochrome in a particular bacterium is correlated with the outcome of an empirical procedure, the oxidase test, which has considerable diagnostic importance in identification of aerobic bacteria. The test is performed by putting a small quantity of bacteria on a piece of filter paper soaked in a dye, either dichlorophenol, indophenols, or N,N-dimethyl phenylenediamine. These dye, which are colorless in the reduced form by oxidase positive species which lack A and C type cytochrome. Cytochrome C. Electron transferred by the cytochromes involved a reversible oxidation of these iron atoms, as shown in equation. In an analog way to oxidation of an iron atom in the iron sulfur protein, the cytochrome have a characteristic absorption band in the reduced state that permit recognition of the several different members of the class, which are designated by terminal letter, for example cytochrome C. Following figure shows the arrangement of electron transport chain in a membrane. As stated, bacteria are quite variable with respect to their cytochrome content. They are also variable with respect to other constituents of electron transport chain. A proposal for the aerobic respiratory chain in E. coli is shown in figure. NADH in the cytochrome generated by a reaction in a fueling pathway transferred on the inner aspect of the membrane two hydrogen atoms to a flavoprotein embedded within the membrane. On the outer aspect of the membrane, the flavoprotein transfers two electrons to an iron sulfur protein, thereby releases them outside when it transfers two electrons to a B type of cytochrome. Finally, the cytochrome B transfers its electron to another cytochrome that is termed as cytochrome oxidase because it has the capacity to transfer electron to molecular oxygen in a reaction on the inner surface that has water as a product. It will be noted that for each pair of electrons transported through the chain, 4 protons are pumped out of the cell, thus creating a proton motive force that can be used to do cellular work. For example, ATP synthesis through the membrane-bound ATP phosphorylation. So students, let us summarize what we have learned in this module. We know that cells are the building block of life. They come in many different shapes and size. Each cell has a specific function to help the organisms to perform tasks necessary for survival. Adenosine triphosphate is found in the cytoplasm of the cell. The cytoplasm is gel-type filler of the cell where all of the organelles of the cell hang out. This location makes sense because the ATP need to be near all the cell part so that it can supply them with the energy they need for chemical and mechanical reaction. Oxidative phosphorylation is made up of two closely connected components. The electron transport chain and chemiosmosis. In the electron transport chain, electrons are passed from one molecule to another, and energy released in this electron transfer is used to form an electrochemical gradient. In chemiosmosis, the energy stored in the gradient is used to make adenosine triphosphate. The energy released by electrons flowing through this electron transport chain is used to transport proton across the inner mitochondrial membrane in a process called electron transport. This generates potential energy in form of a pH gradient and an electrical potential across this membrane. This store of energy is tapped by allowing proton to flow back across the membrane and down this gradient through a large enzyme called adenosine triphosphate synthase. This process is known as chemiosmosis. This enzyme uses this energy to generate adenosine triphosphate from adenosine diphosphate in a phosphorylation reaction. ATP gets its energy by breaking down food. It is created in the mitochondria and powerhouse factory organelle during the process of cellular respiration. The energy is obtained from breakdown food. Animal eat the food and plant absorb the energy from light through the process of photosynthesis. Adenosine triphosphate is considered by biologists to be the energy currency of life. It is the high energy molecule that stores the energy we need to do just about everything we do. It is present in the cytoplasm and nucleoplasm of every cell, and essentially all the physiological mechanisms that require energy for operation obtain it directly from stored ATP. As food in the cell is gradually oxidized, the released energy is used to reform the adenosine triphosphate so that the cell always maintain a supply of this essential molecule. Adenosine triphosphate is remarkable for its ability to enter into many coupled reactions, both those to food to extract energy and with the reaction in other physiological processes to provide energy to them. In animal system, the adenosine triphosphate is synthesized in the tiny energy factories called mitochondria by a process called glycolysis. This is all about ATP generation. Thank you.